Hey all, so today we're going to be looking at HL content related to A3.2, which is classification and cladistics. This is on page 116 to 131, and we're thinking about unity and diversity as our theme. So we want to be thinking about what tools are used to classify organisms into taxonomic groups, and then how do cladistic methods differ from traditional taxonomic methods. So first, we need to know the classification system. So domain, kingdom, phylum, class, order, family, genus, species. Organisms are grouped according to their taxonomy, according to their traits or evolutionary origins, using a system developed over the last 300 years. Higher up organisms, so domain, for example, has less specific traits and more organisms. And then as you go down, they become more and more and more specific. If it evolved from the same common ancestor, it should be in the same taxonomic group. So vulpes here is the fox, and you can see it's in the family of Canidae, canine. And so if we had canines in here, you would expect there to also be dogs and wolves in this group. But they wouldn't be in the same genus or the same species. But if we think about carnivora as the order, we would expect there to be all the carnivores in this order. And so we would expect things that were other than just dogs. So for example, we would have lions in this order as well. This is going to help us to organize a large amount of info, and by viewing certain traits, reduce possible options, so becoming more and more specific. But it can be difficult to classify organisms due to diverging traits, meaning their traits look different over long time frames. And it can be arbitrary categories because there's no objective boundary known whether or not that organism belongs in that family necessarily or not. Or if it's a separate species, for example. So if it has a common ancestor and has similar characteristics throughout that grouping, this is called synaptomorphy. And an example of this would be mammals. And so mammals have hair, they produce milk, they use placentas, and they have a four-chambered heart. And so that's a class. Mammals are a class. And so all mammals in that class are going to have these synaptomorphous traits, and they would be these four. So classification of domains, there are three domains. We have bacteria, archaea, and eukaryota. And this is based on RNA sequencing. So originally, the classification only had prokaryotes and eukaryotes. But then when they looked at their RNA, what they were finding were there were just too many types of different prokaryotes, and they clearly fell into two categories based on their RNA sequences. So eubacteria, that's bacteria, the blue ones here, have peptidoglycan walls. They can cause disease, and they can be affected by antibiotics. So a lot of antibiotics target this peptidoglycan wall because eukaryotes and archaea don't have it. So then it would affect eubacteria, but it wouldn't affect eukaryotes. Archaea tend to live in extreme environments. They don't have a peptidoglycan wall. Usually instead they have glycoproteins or protein cell walls. So typically archaea are highly, highly resistant to antibiotics. They have different glycerol attachments in lipids in the membrane. So they have ester bonds. Ether bonds, not esters. Esters are in eubacteria and eukaryotes. And both bacteria and archaea can have pili, which are little connection devices that allows the organisms to link together, flagella for swimming, and a nucleoid region. But archaea may have histones, whereas eubacteria do not. So when we're thinking about a clade is a group of organisms that have evolved from a common ancestor, either alive or extinct. And the same word for alive, it would be extant. Extant means they're still alive, and extinct means they're now dead. So DNA sequencing of similar genetic bases and similar genetic basis sequences or protein amino acid sequences shows divergence. So If there are more differences, that means they diverged longer ago. So more time, more differences, and this is considered the molecular clock. And we can also look at mitochondrial DNA to trace the matrilineal line because the mom's mitochondria are passed on to the baby. And so you can have a direct line through all of the mothers from the mitochondrial DNA. And so with molecular clocks, this is said to be consistent or assumed to be a consistent rate of change. But this is in fact not, because this can be affected by selection pressures. It can be affected by how long the generation time is. And then also this can be affected by how big the population is, because if you don't have random variation because your population is tiny, you're going to end up with potentially a lot more similarity occurring. And when there's no DNA to synthesize or sequence, morphological traits, which are like physical characteristics, can be used to group species. So every species is in multiple clades. Smaller ones are nested into larger ones. So humans split from our nearest living relatives 4.5 million years ago. And so here you can see Homo sapiens. And then we're in the same clade here as Homo erectus. And then our most recent common ancestor here would be at this clade point, or potentially at this clade point here. But our most recent common ancestor lived about 150,000 years ago. So in order to map the DNA of organisms for us, we can use BLAST and gene banks, and there's links for this in the slides in the speaker notes. And so what we could look at here, and it talks about this in your textbook on page 122, is comparing the genomes of the Philippine tarsier, and the Horsfield's tarsier. And so one of these is in the Philippines, and the other is in Borneo and Sumatra and in Indonesia. And so knowing whether or not these are two separate species could be determined using this tool. And so we can align their base sequences to see how much difference there is. And we can use a distance tree to develop a cladogram for ourselves. And so cladograms, we have these tree diagrams that are showing the most probable sequence of divergence. And further down towards the bottom on the tree means longer ago the split happened. So base sequences can show what order the splits happened. And they're using the parsimony criterion, which is how a species could have diverged with the smallest number of sequence changes. But that's not always going to be true. And so to some extent, there's a little margin of error here. Branch points are called nodes. A node is where a hypothetical ancestor species split to form two more species. So that would be a node right there here. And that would be a node right there. And the root is the base common ancestor. So that would be, in this case, vertebrae. Some are drawn to scale based on estimates of time since the splits happened. So you would have longer lines for longer split times. And some just have numbers that indicate the number of sequence differences. And this, using these cladograms and looking at the relationship in DNA, can provide strong evidence for evolutionary history of a group, but it's not proof. In the 90s, it became quite easy to determine base sequences of DNA. And so as a result, some reclassification happened, especially with plants, due to DNA sequencing. And so sometimes species had analogous structures and they were grouped together when they shouldn't be. And so remember, an analogous structure is structures that are similar because they're well designed and serve a similar function, but they evolved independently. So this is like octopus eyes and human eyes. And other times they were not put with their group because they didn't appear to have the same types of characteristics. And so they didn't always have homologous structures that were similar because of ancestry. So divergent species with a common ancestor may have homologous or analogous traits. And so using DNA is always the best indicator for divergence timeframe and how similarly they are related. So to summarize, there's 3 domains: eukaryotes, eubacteria, and archaea. And taxonomy is used to classify organisms based on most to least similar characteristics. This is domain, kingdom, phylum, class, order, family, genus, species. And you can use an acronym to remember this. Other than domain, I hear King Philip came over for green spaghetti or good spaghetti. And you could pretend like you're telling a doctor that, Dr. King Philip came over for green spaghetti. So cladograms show divergence and evolution from a common ancestor, both extinct or extant, and that may indicate a timeline or a number of different bases. More bases difference means more divergence time, and that can be determined by DNA or RNA analysis of the base sequences of cells or mitochondrial genomes. And so like this is a ginkgo plant, and it's the last remaining species of a clade that arose 270 million years ago. So, you know, they just are almost all gone now. And some restructuring has happened as we have been able to map base sequences using DNA so that we can allow for proper placement of organisms in their correct clades. All right, that's it for this video. Thank you very much for watching, and I will see you in the next one.